Thank you. I thank the organizers for the opportunity to show my work and also visit St. Petersburg, which is quite amazing. I'm talking about a new project. This is the first time I'm presenting it. It was done mainly by Didi Amar, a very bright grad student in my group, together with Herschel Safer. So this is the outline, a brief introduction. The topic is differential co-expression, so I'm going to introduce it, spend some time on how to score this phenomenon, and then describe our algorithm, some results, summary. Okay, so gene expression analysis has been around for a long time, starting from the microarrays in 1995, and more recently the RNA-seq using next-generation sequencing. The experimental outputs are different, but mathematically we get the same object, the matrix. Actually, each experiment just gives one column, which is the profile of an individual tissue or a person. And for each gene, we get a vector of its values across all the experiments. We will use the term, the expression pattern of that gene. So either from microarrays or from RNA-seq, we end up working with the matrix of expression values. Of course, there are important issues of normalizations of the raw data, but we will not address them here. So the classical and the first step in analyzing such a matrix is clustering. So we want to partition the genes into groups, sets of genes. Each color here is a different set, so that each set is homogeneous. The patterns in the same set look like each other, and the different clusters are separated, namely the patterns in different sets are dissimilar. So to do this, we need the notion of co-expression, namely given a pair of genes, two patterns here, two row vectors, we compare them and measure their similarity, typically using Pearson correlation, but could be other ways as well. So we want the co-expression level within clusters to be high and within clusters to be low. One step further is biclustering, in contrast to clustering, which gives a partition or disjoint sets of genes, and you require similarity across all the conditions. In biclustering, we are looking for subsets of both genes and conditions, and we want this co-expression now to be high within a bicluster only on the set of conditions that we are looking. So not across all the pattern, but only on the subset. And this on one hand gives a richer set of possible solutions, but on the other hand, one has to be even more careful in terms of the statistics because the space is much larger. And one of the basic things fields in which this is used is functional genomics. You do your set of experiments, you have a set of genes that you obtain, the cluster or a bicluster, and then you want to evaluate its meaning, biological meaning. And the classical way to do it is by looking at Go classes or other functional gene sets and testing whether your cluster, your bicluster, is enriched for genes from a particular set. And this is typically done using the hypergeometric score, which has an annoying feature that it prefers large classes. So if your set is 50 genes and 70% of them are from the class, or it's 500 genes and 50% of it in the class, you would typically, the hypergeometric score will prefer the latter. But in terms of biology, we always want focused hypothesis. So we would like our gene sets to be relatively small in order that they can be explored by biologists. So it's a common difficulty in this field, and therefore one of the challenges is to come up with methods that will provide relatively compact sets. Okay, as third step in this development, so far I'm describing the progress of the field over the last 2 decades, is to look at data where the classes are labeled. So typically you have a case control experiment, it's the same matrix, but now we know that some of these samples correspond to patients with a particular disease, and the other samples are either a different disease or healthy controls. So this labeling is part of our input. And the basic thing that has been done very successfully is to look at differential expression. Namely, we want to identify genes that behave differently on one set compared to the other. So we are not looking at the whole pattern, but we are looking at the 2 sub-patterns. But unlike biclustering, here the sets of samples that we are looking at are given. And we want to look at genes that are different. So how do we do this? Well, the classical simplest thing to do is to use t-test. Compare the distribution of the values of a particular gene in the 2 sets. When we look at gene sets and not an individual set, we can do something like the Kolmogorov-Smirnov test, or its version that is popularized by the GSEA software. And this works quite well. What we are looking at is differential co-expression. So we are looking at 2-class data, 2 or more, but let's present it for 2. And we are looking at 2 genes. So here are these 2 genes. And where the differential co-expression is the difference in the co-expression levels between the 2 sets. So these 2 genes have to be less correlated in one set compared to the other in a significant fashion. So we are now looking both at a pair of genes and at a pair of classes, and we are measuring this differential co-expression. And why is this interesting? If we find pairs or later on groups of genes that show differential co-expression, the difference may have to do with the disease. So maybe it is a result of dysregulation of these genes. Maybe this is a complex that is working better in one of the situations compared to the other. And indeed, there are good examples where such differential co-expression enabled to identify changes in the regulation. So here is just an example. This is the value of gene 1, let's say this one, and this is the values of gene 2, and the green is the case and the red in the control, and you see that the correlation between the genes is much higher in the controls here than it is in the cases. So, as I said, it can help us to dissect disease processes. So here is a real example. This comes from an Alzheimer's disease case control study. And this is a set of over 100 genes that are much more highly correlated in the patients than they are in the controls. So this is the set of a set of genes, and the differential co-expression can be shown can be observed here by looking at the difference between these two matrices. So this is much bluer than this, so the correlation on the patients is much higher than it is on the controls. Of course, these are the same genes in both. And indeed, if you look at this set, you see a strong enrichment, Go enrichment for cerebellum activity, which is related to Alzheimer's disease. Here is another example from the same study. This is from a lung cancer study. We'll talk about both later. And here you see a more interesting and a little more complex object. So these are actually two sets. This is one set, and this is the other. And what you see is, we'll call this set module, and together we'll call such an object a meta module. And what you see here is that both in the cases and in the controls, the two sets within themselves are highly correlated. You see they are very blue. However, if you look at the interaction between the two sets, you see that it is negatively correlated much more in the patients than it is in the controls. So the differential co-expression here is between the sets. Within each set, there is a high level of correlation, and it's consistent. It is behaving this way both on the cases and the control, but between the set, there is a strong differential co-expression. And just an example, you see these two genes, ALPK1 and RAD23B. This is their correlation in the cases, and this is their correlation in the control. You see that they are virtually uncorrelated in the controls, but they are strongly negatively correlated. And indeed, this is a red point here in the cases. So again, we are looking for object or pairs of objects like this that show strong change in the co-expression between the cases and the controls. And again, there is no particular enrichment that we found for this module, but however, for that module, there is a strong enrichment both for spliceosome and for microRNA preprocessing. Okay, so differential correlation has been discussed in recent years. First in analysis of single genes, later on in finding modules using a profile of one class, so actually using clustering algorithm to find a module in one class, and then testing if there is a differential expression in the other. So find a set in one class and test it on the other. There's also a version of the GSCA that looks at the gene sets, but this assumes that you know the gene sets in advance. You just test your sets in order to find at least one that will be meaningful. The most recent algorithm for this approach is called DIFCO-X, and it detects modules that show change in the correlation, and indirectly it also found module-to-module changes. I'll talk about it later. So we set out to answer a few questions. First of all, when we look at real data, how frequent, how common is this phenomenon? Do we see a lot of differential co-expression? Can it help us really to understand regulation, in particular microRNA regulation? And does it do it better than the classical method, clustering or biclustering or differential expression? Can it assist us in dissecting specific biological complexes or pathways? And if the answer is yes, or actually to answer some of these, we wanted also to develop an algorithm that directly finds a gene set that manifests such differential co-expression. So a particularly difficult or problematic issue in this type of analysis is how to calibrate your data, especially when we are talking in the type of analysis that may be done across multiple classes or multiple data sets. So normalization is very important, and we use the pretty simple-minded approach that seems to be quite effective, although there's a lot of room to improvements. So let R with indices U, V, and D be the correlation between the genes U and V in the class D. And we assume that these values are normally distributed with parameters that depend on the class. And assuming independence between the class pairs, the differences, which are what we'll be measuring, the difference between the correlations for this particular pair of genes in class I and class J will also be normally distributed with the appropriate parameters. And we will use the name t-score for the value for a particular pair of genes and a particular pair of classes, which will be just derived from this formula. When there are multiple classes, so sometimes the data is not 2 classes, but 3 or 5, then we will just take the minimum of these values for, so this is 1 versus all t-score for a particular class. We look for all classes different than our class K and take the minimum value. And if the sign is inconsistent, so we want the value for our class to be the better, if you wish, than with the all other classes, but they have to be consistent. So it be the differential correlation between our class and all other classes should be on the same direction, and we take the lowest value. But we will mainly focus, it's probably simpler to think about the 2-class case as a first attempt. So we collected 6 data sets, class, so 4 of them have, 5 classes, 3 of them are with 2 classes, and the IBD and the lupus data sets have more than 2 classes, typically a couple of hundred samples per test. And they come from different tissues and different types of diseases. And the first thing that we wanted to see is this DC a common phenomenon. So what you see here is, for example, the results on the Alzheimer's disease data. In blue, you see the distribution of the t-score that we computed. And in red, you see the distribution of the t-score on data sets on which we randomized the class labels. So we kept the matrix, and we just randomly permuted the labels of the cases and the controls. And you see indeed that the true t-scores are more broadly distributed than the randomized one. So there is, at the extreme ends of the distribution, we see values that are non-random. And you see the same phenomenon in the other sets. In IBD and SLE, it's very hard to see because, as I said, it's multi-class data, so we take the minimum of several possibilities. But if you look at the end, the tails of the distributions, you see that indeed the distribution for the real signal is broader, and you also see, of course, that our assumption of normality is not satisfied, and even more extremely on the multiple data sets, and I'll get back to this at the end. If you just look at the standard deviation and not at the whole distribution, you see that in all 5 data sets, the standard deviation is larger in the real data compared to the randomized labels data. So indeed, there is, at the ends of the distribution, there is some signal. So we developed an algorithm that we call DICER for differential co-expression for metamodal recovery. And it works as follows. Here is our data set. It can be 2-class or multi-class. So one thing is, one line of study is to compute the t-scores of the real and the random distributions, and to compute sets of genes, we call them DC clusters, of either increasing or decreasing co-expression. We also look at consistent co-expression, namely the classical co-expression across all the conditions, and we will use the combination of the DC and the CC scores in order to compute metamodules, namely sets of genes that are consistently correlated within each set, but that they are differentially correlated between them. And as a side benefit, we also get these red sets from this analysis, although this is not the main goal of the study. And these are what we will call modules, or consistently correlated modules. So this would will behave like classical clusters, but we obtain them only in case they are parts of these metamodule objects. And what we call clusters are the differential co-expression. So the red arcs here indicate the differential co-expression, and the blue arcs are differential co-expression, and the red arcs are consistent co-expression. Okay, so one way to deal with these scores is to use to assume that our data is a mixture of 2 distributions, F1 and F0, F1 with probability P, F0 with probability 1 minus P, and we define the log likelihood ratio as the log of this ratio of observing a particular value S. And again, we assume that the distribution are normals, and then we can come up with an explicit expression of this value depending on the parameters of the distribution. And actually, we'll be using this LLR score in 2 situations. First of all, when we compare genes that are in the same group set, which we call mates versus non-mates, so groups within a set versus group in different sets, and we also use it to compare the real and the random t-scores. So when we have a set of values, so this is now not for a pair of genes, but for a whole group of genes. So suppose we have a set of scores C, and we are comparing 2 hypotheses. One is that all the scores are originating from the distribution F0, and the alternative is that they come from F1. And when assuming that all these scores are IID and they come from the same distribution, then the probability that HI is correct, given the set C, translates just to the sum of their LLR scores. So this idea was used both in the algorithm CLIC that was developed by Roded Sharan and myself, and also by the Samba biclustering algorithm that was done with Amos Tanai and Roded Sharan. Again, we are making some serious assumptions that we know are incorrect. For example, the independence of the value and the normality. But in practice, this seems to be working quite well, and therefore we adopted it in this work as well. So how do we use these scores? When we are looking for a consistent correlation, we will be building the consistent correlation graph. So we now assume, and this is very similar to what was done in Click, we assume that our pairwise values for the correlation of the genes come from two distributions: the distribution of the mates, the genes that belong to the same sets, or the non-mates, genes that belong to different sets. And we use EM to identify, so we are getting a classical example of a mixture of Gaussian, and we use EM to learn the parameters of this value. And the score of a gene pair is its log likelihood ratio score, and if there are multiple classes, we take the minimum. For differential co-expression, we will be comparing the values of the t-scores of the original data and on randomized label data. And we need to set a threshold of when differential co-expression begins, and we just set this prior to be the probability of obtaining the value that is 2 standard deviations more than the normal. So we take a pretty strict threshold in order to take only values that we feel quite confident about. So now we have this prior, this is the P, the probability of obtaining a value from F1 compared to F0. And again, we'll be using the log likelihood ratio score and differentiating the case when the t-score is positive and symmetrically the case that it is negative. Okay, so we will be using the log likelihood ratio score using the randomized distribution as a way to normalize the data. And setting this threshold that will make this admittedly arbitrary choice, but quite stringent of the threshold value. Okay, so how do we find such clusters? So the clusters will be class specific. So now we look at one class, and we want these genes to be significantly more correlated in the class than in the rest, or less correlated. We'll distinguish between the up and down differential correlation. And this will be done by looking for heavy subgraph in the overall graph. And in practice, we used a simple average linkage hierarchical clustering to do this. A more interesting object is the meta module. So again, this is a pair of sets. This is set A and set B, such that within each set, the genes are consistently correlated, but between the sets, they are differentially correlated, comparing the values in the two classes. And mathematically, we can simplify this to the following problem. We have two edge-weighted graphs, G and G prime, on the same vertex set, different edge sets. And a meta module will be a pair of disjoint vertex sets, S and T, such that the sum of edge weights of S and T in the first graph will be positive, and the sum of edge weights between the sets will, in the second graph, will also be positive. And the goal is to find a meta-module of maximal cardinality, the sum of the sizes of the two sets. So actually this is a hard problem. It's also even a hard problem to approximate. We can show a gap-preserving reduction from maximum clique. I think I'll skip this part. And come up to a heuristic that we have developed for the meta-module finding. So those of you who have come across the work on genetic interaction data will feel familiar with this object of a meta-module. And indeed, it comes up in the work that was done in Trey Idecker's lab and later on in my group with Igor Ulitsky. So you're looking for two sets of, so again, you have two graphs. In the genetic interaction scenario, each, so one graph is of genetic interaction and the other graph is of protein-protein interaction. And you're looking for a set of objects that are separately reached in one type of interactions, and between them they are rich in the other type of interaction. And so the input here is this set of two graphs, and the algorithm is a heuristic that starts with a pair of nodes in the graph that have strong differential co-expression between them. And now we take the local neighborhoods of each of these two genes, and this is our starting object. For such an object, we first of all discard nodes that are connected to both U and V. I remind you that what we want is two sets so that there will be many blue edges between them, but many red edges within each set. So we apply several filtering steps in order to clean this set. So first of all, we remove this guy that was connected by blue edges to both. And then we check the red and the green degree, or actually the red and the green weight of each of those particular nodes. And we apply a set of heuristic steps. I think I'll skip this part too. And eventually we end up with a set that we will call a seed. This will be our initial meta-module. And then we do a local, so what we described here is this local neighborhood analysis step. Now at the end of this step, we have a collection of meta-module seeds, and we allow merging of such meta-modules if they match in the colors, if you wish. And then we allow both insertions and deletions of individual genes in order to end up with the final meta-modules that we have. So this is, as I say, at least in this formulation, a hard problem, and the approach is heuristic, and of course there could be other or better approaches as well. So to summarize, again, we compute the differential correlation using the t-scores, come up with the DC clusters and the meta-modules, which are using also the co-expression values, and at the side we also get the consistently correlated modules. Okay, so in order to see how this works in practice, we compared it to diffcox, the most recent method, the co-express, and also a clustering algorithm, the clique algorithm developed in our group, on these 5 data sets. And we limited ourselves to sets of at least size 15. Co-express provided none of these on any of the data sets, so we didn't continue with it. And the 3 others did find sets. Actually, both diffcox and clique only find consistently correlated modules, and the way to find meta-modules is by looking at all the possible pairs of modules and looking for differential correlation between them. This is actually what the DIFCO-X authors suggest in their study, and this is what we did. So here are the results. On average, DIFCO-X finds about 4 modules. Dicer finds much more. In terms of coverage of the total gene set, they are about the same. The average size is smaller in Dicer, which is an advantage, as I remind you the point about the hypergeometric and the enrichment. Okay, so how much differential co-expression does each method actually reveal? So for each meta module, we computed the average DC, both on random meta modules of the same sizes and on the real meta modules, and we compared the fold change between them, because maybe a big fraction of this is just random. And indeed, what you can see, so we are looking at the average fold change. As you can see, DIFCO-X hovers around the value 1. So there is no real difference. Sometimes the ratio is even less than 1. So there is no real advantage in the way differential co-expression is found this way, even though we picked only the very best pairs, very best meta modules of this method. I think we took the 2 best upregulated and the 2 best downregulated in each case, while in Dicer, even though we took everything, we got a much higher ratio. This is also true in comparison to the pairs of DIFCO-X and the pairs of click. So we do reveal more DC in our methods. If you look at individual data set, this is the AD, for example, you see that the absolute change in correlation within Dicer modules is shown here in this red solid line, and this between modules in the blue solid line. And indeed, this is shifted to the right, and it's more than you will see if you look at the DIFCO-X modules. And this is even more pronounced here, where there is virtually no change, no difference here between the between and the within in the DIFCO-X, and it is very substantial in the case of Dicer. What about functional enrichment? So in 4 of the 5 data sets, we see the enrichments of Dicer and DIFCO-X. On 2 sets, the enrichment was about the same number of KEGG pathways. In 2 sets, only Dicer found examples, but if you look at the average enrichment factor, the fraction of genes in the set compared to the fraction of genes in the background, you see enrichment, an advantage to Dicer in all 4 examples. MicroRNA enrichment, so what we see here is, remember that we have modules, meta modules, and clusters, and actually the modules are parts of the meta modules. And this, and what you can see here is that modules and meta modules find different things, and the overlap is not complete. So there are microRNAs that are enriched only on the modules, and there are others that are enriched only in the meta modules. The dark blue is the overlap between them. But we do find a substantial number in all cases. DIFCO-X finds almost nothing. T-test and click are roughly comparable to Dicer. Maybe T-test is a bit weaker in 3 of the classes, but it's strong in the other 2. Specifically looking at microRNAs involved in disease, we had data in the MIR2 disease database for 3 of our 5 diseases, AD, NDD, and lung cancer. And as you see, the advantage of Dicer is very substantial compared to the other methods when we are looking specifically at disease, at microRNAs that are known to be involved in that particular diseases. And indeed, we get very substantial enrichments in all examples. NDD is a striking example because all the 5 microRNAs that we detected are known to be related to the disease. I have here 2 examples. Maybe I'll skip one and just talk about the other. So here is a map of the meta-modules of Dicer that were enriched with protein complexes. So each of these sets are a module, and the red edges indicate down correlation between them, and blue indicates up correlation. And these are the enrichment of the functional or of the protein complexes in each of them. So let's focus on these meta-modules. So we have one module that is enriched with ribosome, and the other is with the 60S unit and the NOP56 of the ribosome. And here is a zoom in on these 2 sets. So if we just look at the interactions between them, and this is using the GeneMania software database, you see that this actually is manifest as one single highly interacting complex, which is not surprising because it's all related to the ribosome. But this type of analysis will not allow us to make any subpartition of this unit. However, if we zoom in on these 2 sets, so you see here the edges indicate high correlation, and if you compare the situation in the cases in the controls, you see that the level of correlation is much, much higher in the patients than it is in the controls. So even though it makes, in terms of interaction and single unit, when you look at differential co-expression, there is a very clear partition into 2 subunits. So the 40S elements of the ribosome come up on this side. The NOP56 come up in this side, and this is the part that is dominated by the 60S. And so even though there's strong interaction in both modules, but there is this very significant differential co-expression. And if the colors of the node that you see here is the level of expression, and you see that only the 40S subunit is up in the patients compared to the controls, while the other subunit is not upregulated. So in this and the other example that I didn't have time to show, Dicer detects changes in coordination between 2 subunits of the ribosome that we couldn't detect by just looking at co-expression or by looking at no information about interaction. So it does allow us to find substructures, and with a relatively modest size. So we don't need to go to gene sets of 50 or 100 genes in order to get a signal. We get the signal even though the modules are relatively modest and therefore much more amenable to biological interpretation. So to summarize, there is a substantial amount of differential co-expression in real case-control biological data. even though we looked at the three types of object, most of the signal is found in metamodules, in these pairs of differentially co-expressed units. And this is true not for Dicer, but also in the other methods. We defined a variant of the metamodule problem, showed that it is hard, and developed a heuristic for it. It is giving us improved results compared to classical methods, both for clustering and for differential co-expression. And the main advantage is when we look for focused changes in the regulation, specifically in our case for microRNAs. In terms of future plans, as I said, this log likelihood ratio assumes normality, and we should probably change it so that there will be a mass value at 0, specifically when we are dealing with multi-class data that looks more like the lower example. We have a parallel project together with the brain research unit at one of the hospitals in Israel, and it's nice to see that many of the tricks of gene expression can also be applied to fMRI data, functional MRI data, and we intend to apply this method as well. And our focus was on microRNAs, and we want to expand the analysis in order to look at transcription factors as well. Finally, let me thank my group and my funding sources. Thank you very much. So you mean that we cover more of the genes using sets?
Well, let's say there was a specific code category for simplicity, associated with a particular data set.
Mm-hmm.
If you just reduce the number of samples that you're using, you see sort of coming out earlier.
So we haven't done this test. What we did test is both Go and KEGG categories compared between the different algorithms, and there we do see a change. Actually, I think we probably are going to the other direction by trying. We have in mind trying to combine different data sets for the same disease. So for each of those diseases that we studied, there are now sometimes dozens of case control data sets, and by merging them, because of the reasoning that you are hinting to, we hope that there will be a stronger signal. Of course, there is a difficulty by looking at different batches, different data set together, but we hope that this log likelihood ratio score will be able to overcome this. This was actually one of the motivations for development.